This with regards to the dextrose solution. Okay, I think yesterday I just put down as hypotonic, whereas we know that the dextrose itself is actually a non-penetrating solute, so it should be the same with the osmolarity. So for example, if 2.5% dextrose, it is hypoosmotic, then it is hypotonic, right? 5% dextrose is isoosmotic, therefore it should be isotonic. But although it is initially isotonic, the nature of the glucose itself is not the same as sodium chloride, meaning that once it is in the interstitial fluid, okay, it can cross the cell membrane through the glucose transporters, and it can be taken up and then be metabolized, right? And therefore, at that moment, the glucose would then be changed into the carbon dioxide and water. So you don't really have the glucose. The glucose itself does not stay in the interstitial fluid and in the cells. Therefore, at that point, then it turns into hypotonic. Similarly, if you have a higher concentration of dextrose solution, for example, 10% dextrose, initially it is hypertonic because it is hypoosmotic and it is non-penetrating, so it is hypotonic. But then once it is metabolized, depending on how much the body the cells take up the glucose, okay, it will then slowly be changed into isotonic and then probably hypotonic afterwards. Okay, so sorry for the confusion from yesterday. Right, so let's start today's lecture. Today's session on the acid-base balance. Right, okay. So the objective for today's session is quite simple lah. But I'm gonna focus more on the physiological part rather than the clinical part. But I'm sure you're more familiar with the clinical part, meaning that how do you interpret the acid-base disturbances and also the type and also the compensation. I'm sure you are much more familiar with that. But my focus for today is to describe what are the physiological changes that occur in the body if the body faces changes in the pH of the body itself. But before that, we need to know what is the normal body pH and also why it is very important to maintain the normal pH range. Okay, so our normal body pH is between 7.35 to 7.45. The pH is actually depends really on the concentration of the H plus or the hydrogen ion, sometimes called as protons as well. So the higher the H plus, the lower the pH. Anything below 7.35, we call it as acidosis. Anything above, we call it as alkalosis. So acidosis and alkalosis really describe the pH of the whole body, the general pH of the body itself. But when we really in the clinical practice, most of the time, the pH that we refer to is the pH of the arterial pH when you take the ABG. So the correct term for the pH of the artery is really acidemia and alkalemia. However, these two terms actually reflects the body pH as a whole, because the H plus itself, when you know the different compartments of our body, they will try to maintain the homeostasis, meaning that if some part of the body, for example, if the artery has too much H plus, and of course it will then be try to buffer and distribute the H plus to the other parts of the body as well. So really, although we only measure the arterial pH, it really reflects the body pH. You need to know that your intracellular fluid, which is the water in the cells itself, has a much lower pH, and of course the pH of the venous end of the capillary or the venous part of the vascular system, it is much more acidic as well. It is at the lower end of the normal body pH. Okay, why do we need to maintain this narrow pH range? Okay. Most importantly, this pH, the presence of the H plus itself can affect the protein structure. So if you remember the protein structure, we have the primary, secondary, tertiary, and quaternary. for the secondary and tertiary quaternary structure, where these amino acids and also the polypeptides, it falls onto each other and forms the covalent and also the hydrogen bonds, that really actually depends on the charges of the amino acids, and these charges can be affected by the H plus, therefore the pH. What happens is that if there's any changes to this pH, it can affect the charges on these amino acids and also the polypeptide, and it can break the hydrogen bond and also causes denaturation. Means that the original form, the original shape and structure of the protein will change. It may becomes it may change from the quaternary down to the tertiary or the secondary structure. So why is this very important? Because most of our important physiological or even anatomical structure in the body, it really depends or made up of proteins. For example, you have your receptors, you have your hormones, you have your iron transporters, the channels, the ion channels, and the function of all these proteins really depends on having that stable structure itself. Other importance of maintaining the pH is, for example, to maintain the oxygen delivery to the tissue, because this pH itself can affect the oxygen hemoglobin dissociation curve. This is called the Bohr effect. I hope you get to learn this with Dr. Rosina earlier. The Bohr effect mentioned that when your body is in the acidosis or when there is a decrease in pH, this oxygen hemoglobin dissociation curve will shift to the right, which then affects or lower down the affinity of the hemoglobin for the oxygen. Lower down the hemoglobin affinity for the oxygen. It's also important to maintain the electrolyte distribution, because H plus itself is cation, as I mentioned before. Our body will try to maintain this electroneutral state. Therefore, if we have too much H plus, of course, they will in one compartment. If you have too much H plus, then you have less of the other cations and probably more of the other anions, just to neutralize the charges. It's also important for the calcium distribution, as well as it has some function in determining the nerve and also the muscle excitability. So these are the details on examples of how the pH may affect the potassium distribution, the calcium ionization and also distribution, as well as the nerve and the muscle excitability. So what is acid and what is base? Okay. Well, the H plus itself is an ion which actually affect the pH. Where does this H plus comes from? So this H plus is usually it comes from the acid. So what is acid? The acid is a substance that can donate H plus. Okay. And then you have two types of acid, which either strong or weak. Or what does it mean by strong? Meaning that the strong acid has the ability to dissociate into H plus and another component or another ion completely. Meaning that if you have a beaker full of hydrochloric acid, what the beaker actually contains is actually a beaker of H plus and a beaker of chloride ions. Okay. But for weak acid, it may not necessarily dissociate into H plus. It may maintain or exist in its original form. For example, this carbonic acid. So this carbonic acid, it may change into H plus and bicarb ion, or it may retain its original form as the carbonic acid. Similar to base, the base is opposite to the acid. Instead of donating, it is the one that is accepting H plus. A strong base is is a substance that readily accepts H plus and convert it into another form. For example, you have the sodium hydroxide, the potassium hydroxide, whereas the weak base, it may or may not accept H plus in the presence of the H plus. For example, the bicarbonate ion, it may form the carbonic acid, or it may just remain as the bicarbonate ion, similar with the ammonia. So because we're talking about the H plus, so we know the H plus comes from the acid. Where does the acid comes from in the body? So we have two types of acid in the body. The first one we call as a volatile, and second one is the non-volatile or the fixed. This volatile is mainly from our respiration, meaning that it can convert it into gases and be released. Okay, so carbon dioxide mainly comes from our internal respiration. And the non-volatile or the fixed acid, it may come from several sources. Okay, most importantly is the catabolism of your proteins or your amino acids in the body, or in some pathological states. For example, diabetes and hypoxia, your body may also produce extra fixed acids. Or in cases of ingestion of other acids, for example, aspirin, methanol, or ethylene glycol. Of course, you have also the source of acid from your daily food intake. Right. So because our body needs to maintain that very narrow range of pH, therefore our body will also have its own defense mechanism to resist against these changes in the pH. So what are the defense mechanisms? So the first one is we have the chemical buffering in the body. So say if you are, for example, if you are eating some lemons, okay, so that is acid. So the body will use this first defense of mechanism, the chemical buffering, to neutralize those acids. But when this is exhausted, then only the body will turn into the compensatory responses. And for the compensatory responses, we have two, which are the respiratory and the renal. The first line of defense of the compensatory response is actually the respiratory, because this respiratory, it can have a very quick effect within 2 to 3 minutes, whereas for the renal compensatory response, the effect can be seen much later. It may take hours to days. However, you need to know that although this respiratory is a much quicker, it has a much quicker effect, and the renal has a much later effect, but the renal is a much more sustainable way, and it is the most effective responses in defending our body towards any changes in the pH. So what are the chemical buffering system in the body? So the most important one is the bicarbonate, okay? And then we have two others, which are the phosphate and the protease. But bicarbonate is the most important one. And the bicarbonate is mainly in the ECF. So it mainly buffers the H plus in the ECF. And where does this bicarbonate came from? It is usually from the kidney. Either it is reabsorbed in the kidney, or the kidney actually generates this bicarbonate. In the ECF, another buffer system is actually the phosphate, specifically the inorganic phosphate. In the ICF, in the cells itself, there are two buffering systems as well, which is the organic phosphate and also the proteins. How does the protein buffer the H plus? As I mentioned yesterday, these proteins are usually negatively charged, so they can usually bind the other cations, including the H plus. Right. Okay. How does the compensatory system works? Okay. Let's look at the first defense, which is the lungs. So this lungs, it usually changes or alters the amount of carbon dioxide expired. Okay. So this is usually done through the changes in the ventilation itself, either the tidal volume or the respiratory rate. How about the renal? Okay, so the renal is also known as the metabolic compensation. It takes a longer duration for the effect to take place, but it is the most effective pH regulator. How does the renal do that? Either it reabsorbs the bicarbonate, or it secretes. It changes the amount of bicarbonate reabsorption and the amount of H plus secreted in the kidney itself. So let's first look at the respiratory regulation of the acid-base balance. Okay, so usually in the respiratory regulation, the pH can be adjusted as quick as within 1 to 3 minutes. So for the carbon, so the respiratory regulation of acid-base balance mainly involve the carbon dioxide, specifically the arterial partial pressure of the carbon dioxide. So you need to know this equation here. How does the carbon dioxide is changed into H+. So carbon dioxide will combine with water, and this reaction will be catalyzed by this enzyme known as carbonic anhydrase to form the carbonic acid. And this carbonic acid is a weak acid, therefore it may partially dissociate into bicarb and also H+. Okay, you need to know that this carbonic anhydrase, it affects this part of the equation, not here. Okay, so it catalyzes the first part of this reaction. So how does the changes in the arterial carbon dioxide partial pressure affect the respiration? What happens is that this carbon dioxide in the artery will then affect the carbon dioxide in the CSF, which then alter the pH in CSF. Okay, and this alteration in the pH in the CSF will then stimulate the central chemoreceptor. These central chemoreceptors are located in the medulla, and from this central chemoreceptor, it actually generates most of the response towards increasing the ventilation. Okay, 70% response comes from the central chemoreceptor. Of course, the carbon dioxide itself may also stimulate the peripheral chemoreceptor, which is the aortic and also the carotid chemoreceptors, but at a much smaller portion, which is about 30%. So what happens is that the central chemoreceptor will then stimulate the dorsal respiratory group, which then stimulates the inspiratory muscles to increase the ventilation, therefore increasing the amount of carbon dioxide exhaled. And this will then return the arterial pH and also the carbon dioxide partial pressure back to normal. Okay, you need to know that this is just a side information. How does the what actually stimulates the chemoreceptor? It is actually the pH of the CSF. Because why? The carbon dioxide itself cannot stimulate the central chemoreceptors. Because this carbon dioxide, it cannot cross, the carbon dioxide, it can cross the blood-brain barrier, but the H+ cannot. Meaning that even if you have a high H+ in the capillary or in the arterial, this H+ itself cannot reach the central chemoreceptor. So it is the responsibility of the carbon dioxide to actually travel into the CSF and then stimulates the production of the H+, which then alter the pH in the CSF. And this H+ can actually crosses the brain CSF barrier here to actually stimulate the central chemoreceptors. Now let's look at how the kidney does it work. Okay, so the kidney, it can do two things. It can reabsorb the bicarbonate and also it can excrete the H+. And the H+ can be excreted in two forms, as the titratable acid or as the ammonium. Yes, ammonium. Okay, let's first look at the bicarbonate reabsorption. So the bicarbonate reabsorption mainly occur in the proximal tubule, and 99% of the bicarbonate should be actually be reabsorbed in the proximal tubule itself. So what are the mechanisms or how are this bicarbonate being reabsorbed? So mainly if you look at the renal, you will have the luminal membrane and also the basolateral membrane. So on the luminal membrane, also known as the apical membrane, there is this important exchanger protein known as the sodium hydrogen exchanger. What it does is that it exports out one H+ and reabsorb, it brings into the cell one sodium. So for one H+ out, there will be one sodium in. And what happens in the lumen is that when this H+ is excreted out into the lumen, this H+ will then combines with the bicarbonate, okay, that has been filtered from the glomerulus, okay? And then it forms a carbonic anhydrous, and then it will be exchanged back into carbon dioxide and water. And this carbon dioxide and water, they are permeable. They can cross the plasma membrane, and so they can actually go back into the tubular cells. And in the tubular cells, the reverse of this reaction occur. So finally, it will actually generate one bicarb and one H+. And this bicarb that is being generated in the proximal tubular cells will then be reabsorbed into the blood through two important carrier, which are the sodium bicarbonate co-transport. Okay, here sodium and bicarb, they are both are transported out, or the bicarbonate chloride exchanger, meaning that one bicarb goes out, one chloride goes in. The bicarbonate reabsorption in the proximal tubule is affected by several factors. The higher the filtered load of bicarbonate, of course, the higher will be the bicarbonate reabsorption. However, you need to know that there's also this reabsorption of the bicarbonate ion has its own threshold or its own transport maximum, which is about 40 millimole per liter. So if your plasma bicarbonate is more, it reaches 40 millimole per liter, that is when the reabsorption is maximum. If the bicarb goes beyond 40, okay, then some of them will not be reabsorbed and some will be excreted in the urine. What are the factors affecting the bicarbonate reabsorption is that if you have volume contraction, so ECF volume contraction, it can actually increase the bicarbonate reabsorption. And of course, if you have more higher partial pressure of carbon dioxide, meaning that it's more acidic, then the bicarbonate reabsorption will also be increased because it needs to neutralize this H+ from the carbon dioxide. Okay. And the next function of the kidney is to excrete the H+, either by as a titratable acid or as the ammonium. So what does it mean by H+ excretion as titratable acid? Meaning that this H+ will be excreted with the urinary buffers. What are the urinary buffers? Okay, one of it is actually the inorganic phosphate. So where does this inorganic phosphate in the lumen comes from? It comes from the filtration at the glomerulus, meaning from your blood, it is filtered into the glomerulus, so it goes into the lumen. Some of this phosphate will be reabsorbed, but some will not, so they may remain in the tubular lumen. So what happened is that in the later part of the tubule, so you know you have the proximal tubule, and then you have the loop of Henle, and then you have the distal tubule, and then the collecting duct, right? So in the part of the late distal tubule and the cortical collecting ducts, this is where the acid will be excreted as the titratable acid. So what happens is that in the cell, again, everything starts in the cell will convert the carbon dioxide and water into H+ and bicarbonate. So this H+ will then be secreted out into the lumen by either the H-ATPase or the hydrogen potassium ATPase, and this H+ will then combine with the inorganic phosphate and to form the phosphoric acid, which will then excreted. So for every reaction in which one H+ is excreted or secreted out, there will be one bicarbonate that will be generated, and this bicarbonate will of course be reabsorbed into the blood. Okay. Sorry. This reaction occurs in the late distal tubule and the cortical collecting duct, specifically in this cell that you need to know, the alpha intercalated cells. So what are the alpha intercalated cells? Okay, so in this part of the renal tubule, you have two important cells, the principal cells and the intercalated cells. So these intercalated cells is the cells that are important in the acid-base regulation. So these intercalated cells, it can appear or it can change the form from type A or alpha into type B, which is beta, or it can also be non-A or non-B. In acidosis, these intercalated cells will be the type A, and that is when it carry out the function that I mentioned before, which is to secrete H+ and reabsorb bicarbonate. But in alkalosis, these intercalated cells will then convert into the type B or the beta type. And then it also has a reverse function. If you look at the diagram here, this is type A, this is type B. The important difference is that the location of these carriers. For example, in type A, in acidosis, you have the you have this H ATP and potassium hydrogen ATPase on the tubular or the apical membrane, and you have this bicarb chloride exchanger, also known as pendrin, on the basolateral membrane. But in alkalosis, this will switch places. Okay, so instead of staying on the apical side, it is staying on the basolateral side, the H ATPase and his the hydrogen potassium ATPase. The H+ excretion as the titratable acid, as a phosphoric acid just now, is affected by the amount of the available urinary buffer and the type of the urinary buffer. So the more the inorganic phosphate is available, of course, there is more H+ that can be excreted, and there are several other urinary buffer that exist. But in my lecture today, I focus on only the inorganic phosphate. Why? Because inorganic phosphate is a much more effective urinary buffer because it has a larger pK compared to the other urinary buffer. For example, creatinine. If I have time, I will talk more about the pK after this. Okay. All right. Another form of H+ being excreted is actually as the ammonium. Okay. To be excreted as the ammonium, there is this concept that you need to know, which is called diffusion trapping. So this ammonium, it can change because the ammonia itself, the NH3 is a weak base. Okay, so it can accept one H+ to form the this ammonia is a weak base. It can accept one H+ to form the ammonium, right? But this ammonia and ammonium, it has a different solubility in lipid. Because of that, this ammonia, it can easily cross the cell membrane, but not the ammonia. So that is the advantage of this, is that it leads to this diffusion trapping, which will help for the excretion of this ammonia. So where does this excretion of H+ as ammonium occur? It occurs in mainly in the proximal tubule, but it can also continue to occur in the process may continue to occur in the thick ascending limb of loop of Henle and the collecting duct. So, but the important one is in the proximal tubule. What happened in the proximal tubule is that in order to generate this ammonium, it comes from the catabolism or the metabolism of this amino acid known as glutamine. So this glutamine will be metabolized by the enzyme known as glutaminase, which produce one ammonium and one glutamate. And this ammonium will be, because it cannot cross the plasma membrane, it will be converted into the ammonia and also the H+. So the ammonia can easily diffuse out, and the H+, of course, needs the protein carrier. And once both of them are out, they will then combine again to form the ammonium in the tubular fluid. Okay, what happens to the glutamate? So this glutamate will then be converted into alpha ketoglutarate, then into the bicarbonate, and this bicarbonate will then be reabsorbed. Okay, what happened to the ammonium in the tubular lumen at the proximal tubule cell? So this ammonia can either be excreted, meaning that it stays in the lumen and will be excreted out into the urine, or when it reaches the thick ascending limb of loop of Henle, it may be reabsorbed back by this special transporter known as the NTCC, or sodium potassium chloride cotransporter. It is being reabsorbed back into the medullary interstitium. What does it do there? It actually participates in the countercurrent multiplication to concentrate the urine. And then after that, although this so it is being reabsorbed, so in the medullary interstitium, it will then be converted back into the ammonia and also the H+. Okay. What happens is that this ammonia from the medullary interstitium, it can cross the plasma membrane, so it can actually reaches the lumen again, and again when it reaches the collecting duct where there is the alpha intercalated cells, this ammonia can actually combine again with H+ to form the ammonium to be excreted. Okay, so these are the ways of how the the H+ can be excreted as the ammonium.
Sorry, doctor. This is Sorry, doctor.
Yes.
Meaning that process happen when the presence of glutamine.
Yes. In the proximal tubule, it needs glutamine.
If there is no glutamine, so this process can't happen lah.
It cannot happen in the proximal tubule, but in the alpha intercalated cells, it can still happen, but then the ammonia will still needs to come from the proximal tubule eventually, right?
Okay.
Yeah.
Okay, thank you.
Thank you. Right. So how or what factors affect the H+ excretion as the ammonium? First, it is the urinary pH. Of course, when in acidosis, you will have more of the ammonium and less of the ammonia, because all this H+ will then be accepted by this weak base to form the ammonium. And also another way in that acidosis affect the excretion of H+ is that the acidosis itself can affect the ammonia synthesis. For example, in chronic acidosis can actually causes adaptive increase of ammonia in the proximal tubule from the glutamate metabolism. Okay. And then hypokalemia means that low potassium can also increases ammonia synthesis. And of course, if you have more ammonia, then you can have more of this H+ excreted. Right. So in terms of which is the main type of excretion of H+ excretion in the body. So in us healthy people, we have approximately the same ratio of excretion as titratable acid and also as the ammonia, slightly higher as ammonium. In patients with, for example, patient in diabetes, what happens is that they in diabetic ketoacidosis, assuming that they have a normal renal function, okay, so they can still increase the amount of H+ excretion either by titratable acid or by ammonium, but mostly it will be as the ammonium, meaning that the kidney will need to generate more of the ammonia to help to excrete this H+. What happened in DKA is that there are more fixed acid production, the ketone. But in chronic renal failure, even if the patient has a normal fixed H+ production, because they have impaired renal function, they cannot really excrete all the H+. There will be reduced excretion of H+ either by the titratable acid or by the ammonium. Why? Because obviously they have impaired ammonia synthesis, which impair the product excretion as the ammonium, and also when they have lower GFR, they will be reduced filtered inorganic phosphate. Therefore, there will be less of the urinary buffer in the luminal filtrate or the tubular filtrate. This is just showing you the important values of the arterial blood gas. I'm sure you're very familiar with this. Some ABG, they also give you the values of other electrolytes, for example, sodium, potassium, and also chloride. The normal range here really depends on the machine that you use and also the lab that you refer to. So in terms of the plasma itself or the blood itself, there is this concept that is known as the plasma anion gap, and this anion gap really depends on the principle of the electron neutrality. It means that your body should have almost the same amount of cations and anions so that there won't be too much positive charges or too much negative charges in the body itself. How do you calculate the anion gap? Usually you take the sodium, you minus the other anions, which is the chloride and bicarb. If you look into some books or some of the medical websites, they also include the potassium here. We call this electrolytes here as measured electrolytes. We have measured cation, measured anions as well. This anion gap really refers to the unmeasured anions that are not balanced by cations. And this anion gap, it really they are the unmeasured anions that are not balanced by the cations. And the anion gap can also be altered by the albumin and also the serum phosphate. So importantly, if you have a patient who has hypoalbuminemia, you need to be aware that the anion gap from this calculation may not be accurate, because in hypoalbuminemia, the anion gap should be much the anion gap should be much lower compared to what you calculate actually from this one. Okay, so I'm sure you're very familiar with this as well. In simple acid-base disturbance, when I mention simple, means that we assume that there's only one source of disturbance, either the metabolic or respiratory, and of course, with every disturbances, you will have to know if there is any compensation from the other organ system has occurred or not, and if it has occurred, is it full or partial? This is just a very much a revision to you how to integrate or interpret the ABG. Most importantly, you need to remember that an ABG on its own does not mean anything. You need to integrate the clinical picture as well as the other lab findings to reach the diagnosis. Okay, so first, of course, you look at the pH. If pH is less than 7.35, it's acidosis. If it's more than 7.45, it is alkalosis. So let's first look at the acidosis. So acidosis can either come from the respiratory or the renal. So respiratory is usually by carbon dioxide retention. So if it is carbon dioxide retention, high CO2, you know it is a respiratory acidosis. How do you know if there is any compensation has taken place? You look at the other parameter, which is the bicarbonate. So if the bicarbonate is normal, means that your body has not tried to reabsorb or generate more bicarbonate. So you say that there is no metabolic compensation taking place. But if the bicarbonate is high, then you know that our body has been trying to compensate that respiratory acidosis. However, you need to remember that for this metabolic compensation to take place, it does not occur within 2 to 3 minutes. Okay, so in acute respiratory acidosis, you probably won't see any metabolic compensation. But in chronic respiratory acidosis, then you may see some metabolic compensation. Another arm of the acidosis is when you have low bicarbonate. So you have you have metabolic acidosis, meaning that your kidney has less ability to regenerate or reabsorb the bicarbonate. So in metabolic acidosis, our body will then try to remove the other part, the volatile acid, which is the carbon dioxide. So usually by increasing the ventilation, so increasing the tidal volume or increasing the respiratory rate, which will then leads to low carbon dioxide. This is when we call the respiratory compensation has taken place. How do we know if it's partial or if it is full compensation? So usually if the pH is abnormal, but there is respiratory, if there is compensation taking place, it means that the compensation is partial. If it is fully compensated, this pH will then go back to normal. So caution, if you look at the ABG and the pH is normal, you cannot straight away say that there's no acid-base disturbance. But it is only normal, or there's no acid-base disturbance if the pH is normal and the other parameters, the carbon dioxide and the bicarb, are also within the normal range. So if you have normal pH, but then the carbon dioxide and the bicarb is not within the normal range, then you probably have a fully compensated acid-base disturbance, or there is a mixed disorder. And similar to alkalosis, alkalosis is mainly because there's so little carbon dioxide or there's so too much of the bicarbonate. What happens if you see both of this? This is what we call as mixed disorder. Okay. How about if you have, for example, high CO2 and also high bicarb? So you probably, if it matches each other, you probably have a normal pH as well, right? So that is also when we call it as a mixed disorder, meaning that you have a lot of carbon dioxide and also a lot of bicarb, or too low of bicarb and also too low of carbon dioxide, which actually leads to a normal pH. However, it is actually a mixed disorder. All right. So let's look at just the simple. So the simple acid-base disturbance, we assume that there's only one organ that is affected and only one compensatory method by the other system. So in respiratory, the primary disorder is within the carbon dioxide. So for respiratory, and the compensation will be in the bicarbonate. So you know, for respiratory, usually the direction of the arrow will be the same. So increase in CO2 and the compensation will be increase in bicarb, similar to alkalosis. But in metabolic acidosis, all of the direction will actually be the same, meaning that the pH will be low. The primary disorder is that something is low as well. Of course, if it is metabolic, then it will be the bicarbonate. And also, of course, the compensation will also causes hyperventilation, which is lowering of the carbon dioxide. This is just a very, I don't know, probably for undergrad, it will be much more helpful for them. But I'm sure in the clinical setting, you don't really need to go by this kind of approach lah, because usually for acidosis, you know pH will be low. Respiratory, you straight away think about carbon dioxide, and you know carbon dioxide is acidic, so you can actually derive that yourself. Right. So let's look at the causes of the metabolic acidosis. Why does it is important to learn about the anion gap? So metabolic acidosis can be divided into the metabolic acidosis with wide anion gap or the normal anion gap. So in wide anion gap is that you have more H plus, either by ingestion or H plus generated from the pathophysiological causes. For the metabolic acidosis with the normal anion gap, it is usually because of decreased bicarbonate. I'm sure you probably came across some other mnemonics for the causes of metabolic acidosis with wide anion gap, but these are the ones that that I use as an example here, gold mark. But most importantly, you need to know that with the wide anion gap, it comes from the gain of the fixed acid. So it's either by intoxication, meaning ingestion of the fixed acid, or diabetes, renal, and also hypoxia, which can actually also generates the fixed acid. What are the causes of metabolic acidosis with normal or low anion gap? It is usually the other way around. Instead of gaining of the fixed acid, it is actually the loss of the bicarbonate. And this loss of bicarbonate is usually accompanied by the increase in the serum chloride. Therefore, this type of metabolic acidosis is also known as the hyperchloremic metabolic acidosis. Why is that? Because, as I mentioned, the principle of the electroneutrality, so you lose the anion, so you need to replace it with another anion. The causes, you can simply remember by A, B, C, D, addition, bicarb, chloride, and diuretics. And usually, if you can easily rule out A, C, and D, you will be left with B. So the bicarbonate loss can either be through the GI or through the renal. Okay. How do we derive which is which? That is when you can calculate the urine anion gap. So the urine anion gap, it is a bit different from the plasma anion gap. It takes into account the sodium and potassium and the chloride, but not the bicarbonate. So if you have a normal function of the kidney, if you have a normal function of the kidney, this urine anion gap will actually be negative. Okay, a normal function kidney, you will have a negative anion gap. Okay, therefore, you know that the cause is actually the other system, which is the GI causes. But if the urine anion gap is positive, more than 20, more than 20, it is usually caused by the renal itself, because this kidney is not able to secrete ammonia. Right. So these are just the list of causes of metabolic acidosis. So what happened in the metabolic acidosis? So assuming this is mainly for the wide anion gap metabolic acidosis, so you have a gain of fixed acid. Of course, the first reaction to it is that you have this buffering system in the body. So in the ECF, this bicarbonate will then try to neutralize this acid. Therefore, in this ECF, you will find there will be less of the bicarb, and also the pH will also be reduced as well. And then what happens in the cells itself, because this H plus it may enter the cells, the potassium in the cell will then leave in exchange with the H plus. Therefore, you may see hyperkalemia in metabolic acidosis. Okay. Once the buffering is exhausted, but the pH is still acidic, that is when the respiratory compensation takes place. So, of course, hyperventilation and blowing off as much carbon dioxide as you can. After 2 to 3 days, then only the renal correction will take place. The renal correction may take place early, but then the effect may only be seen after 2 to 3 days. Okay. So what happens is that the renal will try to excrete more H plus, either as the ammonium or the titratable acid. Respiratory acidosis is much more simple compared to the metabolic acidosis. So respiratory acidosis is really because of carbon dioxide retention. And what are the causes of carbon dioxide retention? It can either occur in the in the main regulation of the respiratory center, which is in the brain. So anything that affects the medullary respiratory center, for example, any medications or lesions in the medullary respiratory center may actually inhibit that center. Oxygen therapy, okay, oxygen itself can actually inhibit the respiratory center as well through the peripheral chemoreceptor. And however, how do I see this? Okay, as I mentioned before, the central chemoreceptor, it is more sensitive to the pH, okay, the pH. But the oxygen itself, the partial pressure of the oxygen itself, it mainly stimulates the peripheral chemoreceptor. Okay. Of course, if you have low pH, this low pH can actually stimulate the central chemoreceptor and to increase the ventilation line. Okay. But then at the same time, if you give too much oxygen, this oxygen, although it may not affect the central chemoreceptor, this oxygen may inhibit the peripheral chemoreceptor, in which it can cause conflict. And if you remember, that is why in COPD patient, you will have to titrate the oxygen therapy very carefully. You cannot give too much oxygen because their breathing, because they have been in a chronic respiratory acidosis for so long, and their breathing is actually the main drive of their breathing is actually the low partial pressure of oxygen. Okay, and therefore, if you give too much oxygen for those with COPD, that may actually remove that stimulus to the peripheral chemoreceptor, and that may actually affect the respiratory drive or respiratory system in those patients. Other causes, so if you think about the regulation of the respiratory system, it comes from the brain, and from the brain it goes down, it gives the information to the respiratory muscles. So anything that affect the respiratory muscles, any CNS conditions may affect that. And then anything affecting the airway or the gas exchange may also causes carbon dioxide retention. So what happened in respiratory acidosis is that you have the buffering, but you won't have the respiratory compensation, obviously, because this is the cause of the acidosis, right? So again, in acute respiratory acidosis, you won't see any renal compensation, but in chronic respiratory acidosis, then you probably see the renal compensation taking place. So next, the alkalosis. Let's see how many minutes we have more. Okay, 5 minutes. Okay, so alkalosis is quite simple as well. The metabolic and respiratory alkalosis, there are not so many causes of it. Let's go to respiratory alkalosis first, because this is probably the most simplest one. So if you lose too much carbon dioxide, then you may have respiratory alkalosis. What is the cause of losing too much carbon dioxide? Mainly it is hyperventilation. For example, if you have hysteria or panic attack, okay, anything that causes hyperventilation. Okay, metabolic alkalosis, it is in opposite to the metabolic acidosis. This time it is because of you lose so much H+, or that you have too much bicarbonate in the body. So example of losing H+ is vomiting and also hyperaldosteronism. Increased bicarb examples is if you ingest bicarb, or something like milk alkali syndrome, or some of the diuretics may also cause increase in bicarb in the body as well. In metabolic alkalosis, yes, you will have respiratory compensatory, but it will be only modest, meaning that it won't be at a higher degree, because hypoxemia itself, meaning that if you try to remove too much, if you try to depress your your ventilation to retain more carbon dioxide, what happens is that apart from increase in carbon dioxide, there will be also low in oxygen, right? And this low in oxygen itself can stimulate the ventilation. So how low is low? It is 60 millimeter mercury. 60 millimeter mercury will stimulate the peripheral chemoreceptor to stimulate the ventilation. Okay, so these are the causes. I think you can read this your own. Okay, I need to talk about the contraction alkalosis. Okay, in terms of the metabolic alkalosis, what happens is that you will have a hypoventilation as the respiratory compensation, so that you can increase the carbon dioxide retention. But the correction can be complicated in contraction alkalosis, meaning that when you lose so much H plus, and at the same time you also lose some water. Okay, so why is that? So for example, in vomiting you lose H plus, so you go into alkalosis. At the same time you lose also water, so you have the ECF contraction, and this contraction will stimulate some other hormones, angiotensin 2 and aldosterone, which will actually increase the bicarbonate reabsorption, which can actually aggravate the metabolic alkalosis. And another consequence of this contraction alkalosis is that you will tend to have hypokalemia as well. Right, so this is the respiratory alkalosis, as I mentioned before. The sequence of event, of course, in respiratory alkalosis, again, only in, this is explosive, right? So in summary, you need to remember that your body fluid needs to be maintained constant at 7.4, and this can be achieved by 3 main mechanisms, which is first the chemical buffer, second the respiratory compensation, and third the renal compensation. The buffering is always the first line of defense, and this buffering is mainly comes from the ECF and also the ICF. The respiratory compensation is the second line of defense by altering the ventilation rate and depth. And finally, the renal or the metabolic compensation is the third line of defense, which actually is its most effective, although it takes some time for the effect to take place. Okay. So thank you very much. Almost 12. I will share this slide with Dr.
Nice.
Yeah. Okay. Yeah. So thank you everyone. Assalamualaikum.
Thank you so much, Dr. Fatin.